Hi, everyone. Welcome to the second day of the seventh RSG Brazil Symposium. Today we're going to start with a talk of Professor Gabriel Valal. Um, Professor Valal, he is a public health researcher at the Fiocruz, Pernambuco, um, and at the Tropical Institute of Bern North of Hamburg in Germany too. He has a master and PhD in animal biodiversity from the Federal University of Santa Maria, and he also has experience in evolutionary biology and genomics, acting in molecular evolution, transposable elements, bioinformatics, and omics methodologies applied to pathogens and vectors. So today he's going to give us a talk that related to um, our theme this year, neglected diseases. It's called Genomic and Bioinformatic Approaches to Study Virus Evolution, the case of arthropod-borne viroses in Brazil. Um, so be welcome, Professor Gabriel. I'm going to put here your
Okay,
talk.
very
So
nice.
whenever you want to start, um,
hmm
stage is yours. Thank you very much for accepting our invitation. And yeah, we can start it.
Yeah, thank you. Thank you, Haisa. So first of all, I'd like to thank you very much, Haisa, and the organizer of this very nice symposium for the opportunity of being here today, talking to you and showing some of the work we have been developing for quite some time now. So as Haisa introduced me, I hope everybody can see my slides in full screen, right? I just did add it to full screen
Yeah,
now.
it's so good.
Okay, thanks. Um, yeah, so today I'm going to talk about genomics and bioinformatic approaches to study virus evolution, the case of arthropod-borne viruses in Brazil. Um, Thanks, Haisa, for this nice introduction, pointing out where I graduated and get my PhD and where I'm located now. But I just would like to give a little bit more background because maybe some of you know me for my work during the COVID-19 pandemic with the Fiocruz Genomic Network. But in fact, I started work with viruses, working with arthropod-borne viruses during the Zika virus epidemic back in 2015-16, right? So at that time, and it's also interesting to give you this context because many of the bioinformatic tools I'm going to talk here today, we started developing back that time during the Zika epidemic. And it's also interesting to highlight that how important is to have sustained funding for research because otherwise we would not be prepared enough to provide and to tackle the COVID-19 pandemics from the genomic and bioinformatics point of view. Um, okay, so now let's then move to the arthropod-borne viruses because otherwise sometimes I speak too much. So today I'm going to talk a little bit about high-impact urban arboviruses, which are the arboviruses circulating in Brazil and the knowledge gaps. How my team and some and with several collaborators, we are filling the gaps using genomics and bioinformatics, and also highlight some opportunities and challenges when dealing with these huge amount of data that now are available, and how we are thinking internally to develop and deploy those bioinformatic tools, uh, focusing on preparedness and maximum impact on public health. Of course, there's also a big deal about the scientific output of what we generate, but we have this inside of my group, we have this in mind of keeping the balance of scientific output, but also how we can use this information rapidly and timely to reach maximum impact on public health. Okay. So, in order to start all this talk, I would like to give you a little bit of background about how high impact arboviruses. So, arboviruses came from the arthropod-borne viruses, which are transmitted by arthropods. But I'm going to talk here today about arboviruses transmitted by mosquitoes. Mosquitoes in general are the main arthropod vectors of arboviruses, although we know some arboviruses that are transmitted by mites and fleas and so on. Okay, but it's important to give you a little bit of context regarding the arboviruses cycles, because we are very well aware about the urban epidemic cycle of arboviruses, because all of you know here in Brazil and South America and other tropical countries around the world that we are highly impacted by dengue virus, by yellow fever virus, Zika virus, chikungunya virus, and so on. But sometimes we forget that those viruses also circulate in other kind of cycles. For example, in enzootic cycles, in which those viruses circulate more predominantly among other hosts like non-human primates, rodents, and birds. And eventually, because humans are approaching those species, these pristine environments where they exist, the synthetic mosquitoes that keep this cycle in the enzootic cycle then eventually feed on humans and transmit those viruses to humans. It's also interesting to point out that many or most of the high impact urban arboviruses that impact human population nowadays, they had and they originated from an enzootic cycle, right? So all of those here, chikungunya does have an enzootic cycle in Asia and in Africa, and now one specific genotype is well adapted for being transmitted by urban vectors and establish this worldwide epidemic cycle. But it also happened for Zika virus, if you remember well. It originated from an enzootic cycle. Yellow fever also has an enzootic cycle in Brazil. And all the dengue, although we do not have much data here because dengue is transmitting in urban settings since a longer time. But we also have evidence showing that every dengue serotype originated from a enzootic cycle. And we also have this epizootic cycle in which also sylvatic mosquito species, they feed on domestic animals mostly. And these domestic animals are the main hosts impacted because they are able to produce enough viremia to infect back a uninfected mosquito, and then the cycle goes on. But again, eventually those mosquitoes, because of the humans are approaching and live in close proximity with domestic animals, then these mosquitoes feed on humans and transmit those viruses to humans, like Japanese encephalitis virus. Um, it's important to mention here as well that for these cases here, for these viruses here, humans are an impacted host, but they do not produce enough viremia to then allow the mosquito to get infected again and then establish a urban epidemic cycle, right? So that's humans here are a dead end host, and the main hosts here that produce enough viremia and keep the cycle going on are mosquitoes and domestic animals. So moving on for the high impact arboviruses regarding humans that impacts the human population, you can see here in this picture a little bit outdated picture that arboviruses, they are more predominantly found in the tropical regions of the globe, all over the globe in the tropical region. Um, but yeah, just a parenthesis here, you're going to see in my slides, in the next slides, that they are spreading um, to temperate regions as well. But here we can see the main arbovirus that impact humans. We have two of them, dengue and chikungunya, are widespread worldwide, right? You can see in all tropical regions here. We also have other like yellow fever, which originated in Africa, but also has a sylvatic and zootic cycle in Brazil. We have Mayarovirus or Pushivirus, Zika virus. It's a bit outdated here because we know there is current circulation of Zika virus in Southeast Asia, and Japanese encephalitis virus. It's also a bit outdated because currently in this year was detected a significant increase in spread of Japanese encephalitis virus in Australia, for example. Here, providing you this background about arboviruses, I will start to visit some of the publications I participated with some colleagues. Here's a paper we published, if I'm not wrong, 2017, where we revisited all the genomic data available for dengue, chikungunya, and Zika in order to try to evaluate if we could find some routes, main routes of transmission of those viruses, looking at the global scale, right? So, and what we found here is that using this genomic success and reconstructing the evolutionary tree, and also using phylogeography, where those sequences were sampled and where the ancestral of those sequences were more likely to occur, then we could found something very interesting. When looking to this South America, more particularly, we found that several dengue virus, chikungunya virus from one specific genotype, and also Zika virus, they spread to the Pacific Islands, and then they arrived in Central America and the Caribbean islands. And this was the local and focal point where most of those viruses arrived multiple times, right? It's important to say it's not. We do have a huge genomic diversity of those viruses, several lineages, not like SARS-CoV-2, right, but with several lineages, and we detected it repetitively, several times on a row of those different lineages arriving through the same main route to the Pacific, right? And after arriving here in the Central America and Caribbean islands, then those viruses spread southwards and northwards. We do have several evidence here. And of course, after this, Brazil, as a big country with a huge population and also a huge population of the vector, became a source for the spread of those herboviruses to other neighbor countries. Okay, so was a review where, yeah, we revisited all the information we had at that time to try to understand, looking at this big picture about virus spread, arbovirus spread. So, going into more detail about arbovirus in Brazil, I would like to give you a little bit more context. So, from what we know, focusing on urban arboviruses, right, those that impact the urban population mostly and are mainly vectored by Aedes aegypti, we do have the first cases of dengue cases registered back in the 1800s, and then some more cases in 1923 in Rio de Janeiro. But after the 1986, there are several outbreaks registered across Brazil. So after this 1986, it looked like the dengue virus really spread across all Brazilian regions and all Brazilian states, and established this local transmission in those states, right? So we do have data for dengue virus, but of course, as you can imagine, at that time we didn't have sequencing capacity. Sequencing um, was developed afterwards, and we only have dengue cases here reported from clinical diagnostics. But if you look at the Brazilian data back to 2001, here we already had some diagnostic tools to monitor dengue infection in the population, although it was only positive or negative cases. We didn't have information about which lineage and more detailed information about the lineages that were circulating. But of course, molecular sequences have been increasing, not only here in Brazil but worldwide for those arboviruses. And from 2001 on, we do have several molecular information available for arboviruses, but only most of the time only from a specific gene, a specific sequence from those viruses. But I'd like to highlight two things with these graphs here. The first one is that you can see these waves of infection every year. Every year, we can see this huge number of cases, dengue cases, occurring in the country. Not differentiating states and regions here yet, but here you can see something different. We had back in 2016, 15, 16, the Zika virus arrived in Brazil. So here we had this huge number of Zika virus cases, right? And here the genomic surveillance, I would say one of the first um, widespread use of genome surveillance was established to study the Zika virus genome during this epidemic, right? Um, but it's also important to highlight here that at that time, what occurred in Brazil was the triple epidemic. Well, if it was triple, then there is another virus. The other virus was the chikungunya virus. Chikungunya arrived probably at the same time, a little bit before, maybe 2000, end of 2014, beginning of 2015, and then uh, at the same time, we had the circulation of three urban arboviruses, right, which spread very easily. They are transmitted by the same highly abundant vector Aedes aegypti. But another very interesting thing after this triple epidemic that you know you probably remember this spread across Brazil, we had this drop in cases here. It's a huge drop. It occurred for dengue virus, for Zika virus, and also for chikungunya virus. So that's one thing my group tried to address in one of the studies. I'm going to show you what happened in this case, why there is this huge drop. Maybe a large part of the population was infected, and then a significant immunological barrier was established, and then in the next year we had a much smaller in scale infection wave, right? And here is just another, the right part of the slide here. You can see the different regions of Brazil and how dengue virus specifically were circulating substantially in all regions of Brazil. Of course, there are scale differences. When you look at the south region, we do see the waves of infection, but they are much smaller in scale compared with the southeast, the northeast, and central west regions. I'm going to talk a little bit more about this later on. Of course, when we talk about arboviruses, we need to also consider several other factors like the vector population, and we need to consider where the cases are occurring, are occurring in Brazil, in order to evaluate if there are any trends, future trends that we need to be concerned about and study those trends. So here in the left Brazilian map, you can see it's quite outdated, right? It's a map from 2007. But here in this map, you can see the infestation of municipalities in Brazil, where Aedes aegypti was already present in a certain abundance, right? And here we do have a more recent or larger time frame, in fact, from 2002 to 2012, reporting dengue cases across Brazil. And two different things emerged from those maps here. We could see clearly that there is a barrier here and another barrier here, right? Most of the Aedes aegypti infested municipalities were concentrated here, and almost very few municipalities were infested in the southern part of Brazil and mainly in the Amazon region. So here is something that's easy to interpret because, as I just mentioned several times, Aedes aegypti is a urban mosquito, urban adapted mosquito. So if we do have a large forest here, Aedes aegypti will not enter or will take longer to invade such region. And there's another explanation. As you all know, our country is mostly tropical, but we also have a subtropical region where the temperature variation is much more demarcated throughout the year. And because of that, Aedes aegypti is not very well adapted to cold winter, and then the winter and more lower temperatures here acted as a barrier, buffering the spread of Aedes aegypti to the southern part of Brazil. Okay, yeah, here is just something to follow up. Here are the climatic classification map from Brazil from 80 to 2016. Look specifically at this tropical region, rainforest, and so on. And here we have a temperate region. And here is another study that was showing the mean temperature variation per month in several years, following years. So you can see that in the three southernmost states of Brazil, Paraná, Santa Catarina, and Rio Grande do Sul, we do have this well demarcated temperature variation. Here we have winter time, and of course this winter time was enough to buffer the spread of Aedes aegypti and consequently to buffer the spread of the virus uh, uh, that could affect the population of the region. Okay, but now I'd like to bring to you some more up-to-date information for specific for dengue virus, but you're going to see that it's not restricted to dengue, unfortunately. Uh, Regarding the dengue expansion in Brazil, considering the 2021-22 period. So this is a recent publication that summarized the dengue cases in Brazil. And one thing that can pop out, maybe you have noticed that neither of the two barriers are really clear anymore, right? So here we have dengue cases all spread to in uh, the Amazonas municipalities. And here you can see that dengue cases were spreading southwards to new municipalities, covering big part of Rio Grande do Sul, Santa Catarina, and Paraná state, right? And another point that this paper raised here is that several of those municipalities here reported dengue cases for the first time in history. They never had any kind of dengue cases in the past, but now they reported the first cases, right? And most of the cases which characterized this specific 2021-22 year, which was just to recall, it was the record of we had the record of cases in Brazil um, compared to the whole history. Most of the cases occurred in the southern part of the country. Why? I'm going to show you a little bit more later, but it's of course a combination of global warming, a combination of vector spread to this part of the country, and also because the population, the human populations there, was not infected in the past and it's completely naive for new infections. So in this part of the country now we have the perfect storm for new outbreaks of these arboviruses. So now moving to chikungunya, which I will also talk about today, give you a little background. Remember that chikungunya arrived in Brazil back in 2014-15, right? And chikungunya is interesting because it arrived at the time we already have the tools to perform genome sequencing and characterize those viruses in more detail. And regarding chikungunya, we detected as a community two main genotypes that arrived in Brazil. The first one was the Asian and Caribbean genotype that had arrived through that specific main route I told you about in the previous slides, through the Caribbean island in Brazil, in the north part of Brazil. And the second genotype arrived directly from Africa in the northeast part of the country at Bahia state, right? And in this same paper, the authors try to model it and to predict if those two genotypes would be able to spread countrywide. And of course, due to the climatic conditions and the spread of the vector population in basically all municipalities in Brazil, they yes predicted that those two lineages would spread eventually. would co-circulate in some regions of Brazil. Okay. Okay, so before moving on to some of the findings we have been generating inside of my group and in collaboration with several other scientists from Brazil, I'm going to talk a little bit about the bioinformatic tools we have been developing to really map the spread of those viruses in a timely way, thinking about how um, we can sequence the viral genome very rapidly from a significant number of samples that came from that's geographically representative and time and also representative in terms of time frame, and then with these genome sequences implement some analysis to extract insights that could help us to fight those viruses. So here I'm going to talk a little bit more about the genomic surveillance engine. That's something we came up as an idea during the beginning of the COVID-19 pandemic. But again, as I pointed out before, some of these tools were delineated back in 2015-16. So the genomic surveillance engine, we thought about how we could set up some tools or some pipelines that where we could enter genetic data, genetic information from any kind of pathogen. Of course, you're going to see more about viruses, but the idea is more general than that. And then we could perform systematically the same analysis, genome assembly, analysis, mutational analysis, and some phylogenetic analysis. And in the end, we would end up with a bunch of information that's highly relevant for scientific purposes, but also highly relevant in terms of genomic surveillance, of timely release of data. And within this genomic surveillance engine, we thought not only about tools and pipelines, but also about databases and systems that could help us to generate standardized outputs, how we could protect the data, this sensitive data of pathogens, and eventually we sequence material from the host, and also how we can share this data systematically and programmatically with the world, right? In order to really have this comprehensive analysis of pathogens. And one of these tools we published it during the pandemic because it crystallized finally during the pandemic, which is called ViralFlow. So ViralFlow is automated workflow for SARS-CoV-2 genome assembly, lineage assignment, mutations, and intra-host variant detection. And when we were developing ViralFlow, we thought about finding a given balance to allow everyone to use it, like running personal computer, or if you do have access to a large computational resources, you can then scale it up using multi-user servers, right? So we provided two ways of implementing it, through manual implementation or through container services. At the time, the first version of ViralFlow, we used Docker images to facilitate implementation, but not only, it's also very important for reproducibility. But you're going to see that we changed this implementation strategy a little bit. But going into more details, basically, ViralFlow, it's reference-based genome assembly, right? So we do have these two main strategies, de novo assembly and reference-based. In the case of ViralFlow, as it's more tailored for viral genomic surveillance, and viral genomic surveillance is basically implemented for viruses that we already know about it. So that's the main concept. We extract and we decide which is the reference genome, and then we generate sequences, genomic sequences from other lineages of the same virus, and then you map against the reference genome to perform all the analysis, right? So it's a pipeline that performs some basics steps like filtering, low quality reads, And then using some mapper tools like BWA and some tools, we generate a final bound file. And from this bound file, we extract the consensus sequence and perform some other analysis. But one thing I'd like to highlight here, instead of going into much detail about all these steps that are also implemented in our pipelines, I would like to highlight one of the differences of Varaflow. So Varaflow, since back in 2015-16, we started to detect some co-infections events, and we also were interested in characterizing the intra-host variability, because as we know, those viruses, they do have a high mutation rate, and every time they replicate within a cell or within a tissue, they generate a lot of what is called a mutant cloud. So not every genome is exactly the same as the original genome, because they do have an RNA-dependent RNA polymerase, which is error-prone. It inserts new nucleotides, new errors in the replication, but that's allowed the virus to explore more the fitness landscape. So we decided to go through and implement some, at that time, a Python script to characterize the what we call minor variants or intra-host variability. Because most of the pipeline only generate the consensus sequence, right? Which is pretty interesting, really important. But there's quite a lot of variation in the minor section, or the intra-host variant. So inside of Varaflow, we can systematically generate minor variants. And because we collected this information for every sample, we also had another idea of building alternative consensus sequences. So there is the major consensus sequence and the minor consensus sequence, right? You can ask me about, well, there are other strategies and other tools that does try to reconstruct this cloud of genomic versions from within the cell. And yes, we knew about it, but one thing is that those pipelines, they are really interesting in terms of scientific analysis, where you do not really need speed, right? Uh, but remember, here we were always concerned about genomic surveillance, and we need to have speedy results. So we decided to implement our own Python script, thanks to Felipe Desordi, which still a PhD student from my group. He implemented those ideas, and then we could generate these minor variants very rapidly, and also generate this alternative consensus sequence. And with this information, I'm going to show you that we can detect some co-infections event we detected for SARS-CoV-2 and also for arboviruses. Okay, and it's also important to mention that the later steps of the pipeline, we implemented some software that were specific for SARS-CoV-2, some analysis that were specific for SARS-CoV-2, but as you know, Nextclade also is implemented for other viruses. So we are still planning how to implement Nextclade for arboviruses. Here's just a performance analysis in different research settings. Remember that we were very concerned of implementing Varaflow in a way that it could be used in low research settings. So we could see here the same data set of 100 samples. We could run it in a laptop in around 30 minutes. For every sample, it takes around between 3 and 6 minutes to run and generate all the outputs, right? But of course, it depends on the amount of reads, the amount of sequencing reads you obtain for every sample. But we showed also that we can scale it up in HPC clusters and then run the same 100 samples in 5 minutes, for example. But of course, we kept developing Varaflow, and now we are working on a final version of Varaflow 1.0, which will hopefully come out in the beginning of, maybe end of this year, beginning of next year, where we changed some stuff inside of Varaflow. So this is the new webpage of Varaflow. It's still in the beta test version, right? but please, if you are interested, you can go to the web page and use it and let us know if you have any issue, because we are exactly trying to debug and finish VaraFlow right now. So when we start developing VaraFlow 1.0, it's also important to thank you here, Antonio Marinho, which now bioinformatician of the Sanger Institute in the UK. He started developing or recoding VaraFlow to include it inside of Nextflow, this domain-specific language that was developed, which is pretty good for bioinformatic pipelines. But also we decided to move to Singularity because we detect some issues with Docker images that it requires some sudo access to run some Docker images. So then we move it to Singularity. Here's the new VaraFlow 1.0. I want to go to the details of the basic steps because most of it was recoding and establishing a new in this new Nextflow specific language. But we included some new tools, for example, SnipF, which not only, so the pipeline with SnipF not only provides information about the mutations, but also can give you some idea if these mutations, if a specific mutation was synonymous or non-synonymous mutation, and then if the amino acid change do have a very high impact or not in your protein, right? So we still have the intra-host script, still have the specific lineages, and we compile all the outputs in a easy way to generate reports. Here I have a summary of the main differences of VaraFlow 1.0. The first thing, and I'd like to highlight here, is that we now have a pipeline that's virus agnostic. It's not complete agnostic because VaraFlow was set up to work with linear virus with linear genomes, right? So for segmented genomes, it does work, but then we need to pass one segment at each time. But we are working on it to maybe allow VaraFlow in the future to work with segmented viruses in a systematic way. But for now, for viruses with linear genomes, as long as you have a gene bank ID, you can pass the gene bank ID to VaraFlow. VaraFlow will download the genome and the genome annotation, and we'll set up VaraFlow for running at least the core analysis and the agnostic models. So which are diagnostic models. Here's one example. The intra-host variant, they will be run for every virus. So this is an agnostic model. The consensus genome is an agnostic model. The major genome and minor genome are also agnostic models. We also implemented other things like generating mapper reads only, because that was a big concern. Because when you think about data sharing, because when we use this kind of amplicon-based sequencing, of course, we are enriching substantially for genetic material of the virus, of the pathogen you are interested in. But in the end, you always end up with some human reads, human genomes being sequenced together, right? So if we share the raw data, then we are sharing not only the pathogen reads, but also some reads from the human subject, which most of the ethics committee approval does not allow, right? Because you asked the ethics committee to analyze only the pathogen data. So with these reads now, only the ones that map it against the reference genome, we are sure that we can share those reads and only the pathogen genetic data will be shared. And we now are working in a specific database to automate the submission of the data to some specific database. Of course, we also have the virus-specific analysis as well. Here's a recent implementation that was thanks to Alexandre Freitas, another PhD student from my group, that implemented inside the VaraFlow the Bandash software. This is software developed by Jonas Fuchs, which allow us to see for every sample the number of reads mapped per position. And this is cool because not only provide us some static figures, but we can also open up some HTML, so we can zoom in, zoom out, and see specifically some regions of the genome. Well, this feature is particularly useful for the wet lab team. Not only the wet lab team, the whole team that works with viral genome surveillance, but for the wet lab team particularly because when there is a dropout, some primer sets are not working, then they can see exactly which is the primer set that are not working with this tool here. And here's an analysis from Felipe Desardi, which extracted some data for a recent talk, and I asked him if I could use this slide. So he extracted from GISAID data. He extracted all the Brazilian data and was looking at which is the most used software for genome assembly and genome analysis of SARS-CoV-2 data. And although almost, well, a little bit less than half of the sequences, there were no indication of the software used, which is not good for reproducibility. If we consider all the other half, Viraflow was the most used one compared all the commercial software. So I think that this was very reassuring for me and for the group to see how Viraflow is being used and how maybe it was easily deployed in a way that people could really use for genomic surveillance and for scientific papers. And the other solution I would like to talk about is Virus Genome DB, which is pretty much linked with Viraflow. You're going to see it. So we call it Virus Genome DB as a database, but in fact, it's a more complete system, right? So Virus Genome DB, we are building it to be user-friendly because many of the users are people from the wet lab that get the samples, that take notes about the samples and process it through the sequencing pipeline. And we would like to make it easy for them to store all the information and even run Viraflow inside of this Virus Genome DB. So Virus Genome DB, we can follow up with data visualization, for example, the lineages that are circulating, the coverage of each sequencing run of different, in the case of Fiocruz network, of different Fiocruz unities that are generating sequences. We can see for everyone, every run, the number of reads mapped against the negative control and the positive samples, and then perform some quality checks here. VirusNodeB was also set up to submit or prepare the data for submission directly to GISAID, not only consensus, but we are working to submit directly the reads, only the reads that map it against the viral genome. And we can download several things, right? We can download all the viral flow output. And one thing that was very, very important in terms of genomic surveillance is that inside of our Genome DB, we standardized the reports. So these reports here are sent to the Minister of Health, to the different sectors of health at the state and municipality level. Then we could easily share the information in a systematic way, in a countrywide way, using this standard report, right? So automatically, Virus Genome DB will extract the information of the lineages and will generate the backbone of this report that still can be edited for some specific purposes. Okay, and here I just mentioned this before. We are using Jenkins, which general orchestrators of pipelines to automate several things. So with Jenkins, the user can not only upload the data, the metadata, but with one click, they will run Viraflow in the backend, will import back the data, and then with another click, Jenkins will launch the submission of the data to GISAID, but only the genomes that reach a given consensus quality. Some side of the network we established at some very high threshold to submit the data to be saved, right? So moving back to the studies using these tools, here is a paper we published, if I'm not wrong, 2017 or 18, where we sequenced the chikungunya virus from samples collected in Pernambuco state. Back there, Varroa was not deployed yet. It was a bunch of bash scripts, right? But that was the original version of Varroa. But the interesting here that, remember, chikungunya arrived in the north and the northeast Bahia state, but Pernambuco in the middle of this, let's say, not exactly in the middle, but between these 2 points. And what we did here is that we sequenced several samples, showing classical chikungunya and also some stream um, um, symptoms of the chikungunya fever. We recovered full genomes and some draft genomes, but the most interesting finding in this paper here is that we detected co-infection and co-infection of the 2 genotypes, right? Remember that I told you that those 2 genotypes could spread countrywide and even overlap in distribution. So with those samples here, we detected co-infection. And how do we detect co-infection? Because we analyzed the major consensus and the minor consensus, and we characterized the intra-host variability. So when we look at the intra-host variability, we can see here every genome position where there was variability. It means sequenced reads supporting one nucleotide position, and reads also supporting an alternative nucleotide with enough coverage, right? So we can see throughout a single sample genome, viral genome, that we have several positions across the genome where we have 2 alternative nucleotides, right? And when we look at specifically, well, does this nucleotide belongs to the Asian Caribbean? It's a nucleotide already found in the Asian Caribbean genomes, or it was already found in the CSA, the African genotype. So most of them were already characterized in one genotype or the other. So that's why we concluded in this paper that we found here co-infections of those 2 genotypes. Of course, we could go further and try to extrapolate that, well, those co-infections are leading to more extreme symptoms, but remember that we had only a small number of samples here, so we cannot really evaluate it with this set of samples. Another study, more recent study we published in Memoirs do Instituto Oswaldo Cruz, was the largest outbreak of chikungunya in the southernmost state of Brazil, right? So remember that chikungunya arrived in the north and northeast part, but very few cases have been detected in the southern part of Brazil. So in this paper here, we provided, in fact, we sequenced the chikungunya genomes from samples from 2017 to 2019, right? And during this time frame, before 2021, the number of chikungunya cases were never higher than 52 in the entire state, in the entire Rio Grande do Sul state, right? But then in 2021, we detected 220 cases, and they were mostly restricted to a single municipality, which is called San Nicolau, from the northwest part of the state, right? So in this year here, the 220 cases was a number higher than summing up all the cases registered before in the state. That's why it was the largest outbreak, chikungunya outbreak in the state so far, right? But then with those genomes, what we can, which kind of insights we can extract from those genomes? So we performed a phylogenetic and phylogeographic analysis using Brazilian states and genomes from other South American countries, and we could detect that all the you can see those arrows here, all the chikungunya genomes detected before 2021, they were derived from introductions, right? From interstate introductions. And some of these cases here, we had the epidemiological information, the metadata showing that was somebody that traveled to those countries, those states where there was clearly endemic circulation of chikungunya, and then it came back to the state and was diagnosed with chikungunya, right? But the important thing here is that even those introductions, they we couldn't detect any further transmission in the Rio Grande do Sul state. So it did not establish a communitary transmission at that time. But then in 2021, we detected then this huge cluster, and of course, some people still were had this link of traveling to endemic area, and that was likely the way the chikungunya was introduced again in Rio Grande do Sul. But now what we saw is that this introduction really triggered the communitary transmission and a large, very large outbreak in that state. Okay, so we used phylogenetic analysis and phylogeography to assess this. Another study is about dengue 3 virus introduction in Brazil after a 15 years gap. And was recently published, led by my colleague, Dr. Felipe Naveca from Fiocruz, Amazonas. He and a large team, including several people from the Fiocruz Genome Network, we sequenced dengue 3, serotype 3 virus. And that's a pretty important finding in terms of genomic surveillance because of this 15-year gap. So in Brazil, before this detection and this publication, only dengue virus serotype 1 and 2 had been circulating for quite a while. But then when a new serotypes arrive after all this 15 years gap, there is a fear, there is a concern that the population of South America in general, but also in Brazil, do not have any or have very few antibodies against the serotype. So we are following very closely this finding here because we are expecting to see a huge increase in cases in Brazil and also in South America due to the spread of dengue virus serotype 3 of this new introduction, right? And it's also interesting to highlight that this introduction occurred again. Here we have Indian genomes from sampled in India and also in Bhutan. And of course, there are plenty of places that probably were not sampled at the time, but we can see clearly that it probably followed with the Pacific route as other dengue lineages followed in the past. Let me see. And this is another paper that was published now that we do have genomic surveillance for arboviruses. We still need to improve it, but we do have it across Brazil and also establishing in South America. We detect this multiple introduction and countrywide spread of dengue 2 genotype 2 cosmopolitan in Brazil. So here we are seeing again, that paper did not show the introduction of dengue 2 genotype 2 because another previous paper showed that, but here with a larger data set and because of this genomic surveillance, we were able to catalog at least 4 introductions from Peru to Brazil, right? And it's interesting because in Peru back in 2019, 2020, there was a huge outbreak. We have almost no data about the serotype and genotype, but we are speculating that, of course, it probably was genotype 2 cosmopolitan was important. This outbreak in Peru that seeded new introductions to Brazil, right? Well, and why these examples, right? I showed you that we do have simultaneous circulation of the serotypes, genotypes, and lineages. And I think that's, I'd like to just come back to that question of various lineages that co-circulate and what's the importance of this. Because you probably remember from the SARS-CoV-2, now the main lineages are recombinants, right? So 2 lineages that recombined and exchanging information and generating a new, more fit lineage. And we also can detect this kind of thing for arboviruses. Well, not the recombination yet, but as I showed you, viral flow is able to characterize and detect co-infection cases, right? Here I brought you an example using SARS-CoV-2 data, where we published a recent paper showing that using the major consensus and the minor consensus, we could see that each one of them were clustered in different lineages, very well supported, right? So if we do have enough markers that differentiate those lineages, and those two lineages were co-infecting the same host in the same cell, we can extract this information and characterize this co-infection from the sequencing data using viral flow. Okay. And the last part of my talk, I'm coming to an end. I would like to talk a little bit about cross-sectoral and cross-country collaboration to tackle arboviruses. So maybe some of you know, but we recently published this letter in the Lancet Global Health journal, where we called for a urgent need for global surveillance of arboviruses, high-impact arboviruses. So we established what's called APRBO DB inside GISAID, where people from across the globe are depositing, are submitting data for public health response. So for now, it's mainly focused on dengue, chikungunya, which are the two worldwide widespread arboviruses with the largest the largest impact on the human population. But we already implemented for Zika and are planning to implement for West Nile virus and yellow fever virus. So here is the front end of the APRBO dengue. Several of the tools that have been used for APCall for coronavirus, but also for other viruses, are already implemented here, and we can perform very rapid analysis of that. So here's the data, recent data from Brazil. We can follow now based on the genomic and not only complete genome, but also fragment data. We can follow the serotypes abundance and replacement through time. And here we have dengue 1 and dengue 2, which still are the main serotypes circulating in Brazil. But dengue 2, you can see here in the lower part, we do have two genotypes, and we are detecting more and more cosmopolitan, two cosmopolitan genotypes. And to finish my talk, I just would like to bring you the attention that we are dealing with global warming, and arboviruses and the vectors are taking the advantage of that, right? So this is a paper where the authors try to estimate the spread and the impact of arboviruses, taking into account the different estimates in terms of global warming in the future. For example, here is just a zoom in the global warming estimate for business as usual. If we do not do anything to contain global warming, and with medium challenge to mitigation adaptation. And you can see here in this kind of heat map, if months suitable for the transmission of those arbovirus are expanding or retracting, right? So if we go to the negative values here, we're gonna have less or shorter periods of time for the transmission of those arboviruses. If we go to the red part and pink part of the graph, then we're gonna have more time, more months that are suitable for the transmission. And you can see here in the southern part of the country that, of course, we do have yellow or orange here, showing that we're going to go for longer and longer period of time that are suitable for viral transmission, which then will allow the virus to transmit more rapidly and cause these larger and larger outbreaks. Yeah, and this is linked with what I mentioned at the beginning of my talk regarding eroding climatic barriers. Here you remember these very well demarcated seasons in the southern part of the country. We still have winters here, but they are not well demarcated anymore, meaning that we do have more average temperatures, and those average temperatures not are very good for the vector, and of course to also transmit viruses in the southern part of the country. So if you look at a change in months suitable, we have an increase in months suitable for viral transmission, arbovirus transmission, particularly in the southern part of the country. That's why we have almost no climatic barrier here to buffer the spread of those viruses. So the take-home message are arbovirus burden in Brazil is increasing due to the circulation of new lineages. Recent increase in incidence is more pronounced in the subtropical region because of the points I raised here. There is a combination of factors leading to this epidemiological change, as arrival and local transmission of virus, lack of immunological barrier, and large population of the vector. Timely genomic surveillance of a virus can provide critical and detailed information to guide mitigation efforts, besides generating new insights. And but to really have timely genomic surveillance, we need to deploy robust and easy deploy bioinformatic tools are required to maximize the impact of genome surveillance on public health. And with this, I'd like to thank you. Of course, all this work is not done by myself alone. It's a big team led by many, many hands. I'd like to thank you very much, all my team and collaborators that are working tirelessly in these last years. And also, of course, thank you all the funders and the institutions that are supporting us to perform this work. With this, I'd like to thank you again for the invitation, and I'm happy to take any questions.
Thanks so much, Professor Gabriel. That's a wonderful talk. I'm not from epidemiology, so I was not aware of the viral flow. So I'm like really impacted. This wonderful work that is being done here in Brazil. So we have a couple of questions in the in the chat. The first one, great presentation. Sorry, great presentation. Thank you. Why was the workflow created using Nextflow? Is there any specific advantage to this?
Yes, so we decided to move to Nextflow because in the past, ViralFlow was a couple of Docker images, and we call it, using some Bash scripts to perform the analysis. But when we decided to study more this domain-specific language, specifically tailored for pipelines, they provide us some very, very good tools, not only for developing the pipeline, but also to use the pipeline. So I am more, yeah, I know more about Nextflow, but also know that there are at least two others. SnakeMake, it's another common specific language for pipelines, and there's another one I can't recall now. But talking more about Nextflow, one thing I like very much of Nextflow is that first, it can integrate any kind of language, computational language. So we still have this Python script. It's easily integrated. We can easily integrate it inside of Nextflow. If we do have an error script, we can call it inside of Nextflow, and it goes on, right? Another thing that Nextflow is pretty useful is that as we set up the models and we set up the processes of the pipeline, we name them, right? As we do for any other kind of pipeline. But inside of Nextflow, when you run it, then you can follow every step of the pipeline. For example, if we do have 100 samples, it's going to show, well, cleaning the data, let's say. It's running the software to remove low quality reads. And then it will pop up the numbers, how many samples are running, and how many samples already finished, and if all the processes finished well, and then it goes to the next one, and the next one, and the next one. And also another thing is that Nextflow works pretty nicely in terms of parallelization. So you can launch several jobs at once if you do have several threads and cores in your computer, and it will handle it by itself. It works pretty well. So I think that was one of the main features that allowed Fireflow to even gain speed when we move it to Nextflow compared with our original version, because In principle, we thought, well, we will include new softwares here, then the pipeline will get slower, and then let's do we should we do it, because yeah, we consider, we always keep in mind this thing about genome surveillance and timeliness. But moving to Nextflow, it even was faster. So I really enjoyed to to recode Varaflow to Nextflow.
Great, great. This takes one little question up from me, because you were talking that any person can go to the website and put the virus sequencing, and then it can find it like what they need. But how long does it take to
So
run? Because you said
yeah,
that.
mhm. Just to clarify, in fact, the website, you'll find the instruction to git clone, to clone the pipeline,
Okay.
and to install the basic
Oh, okay.
dependencies, which we try make to make it very simple, very few dependencies, and then um, all the processes goes along. But when you do have the pipeline in your computer or in your cluster, you can pass the gene bank ID of your
Oh,
given
okay.
virus,
Mm.
right? And then it uses the efetch, one specific script of the SRA toolkit from NCBI, if I'm not wrong. And then it consults NCBI and download the annotation in the sequence. So it's pretty easy, as long as your internet is working well. We already have some issues with that. Sometimes the internet is not very stable, but yeah, it
Okay, so
works
it doesn't
very
use
well.
much memory from the computer. And
No, no,
okay.
not at all, not at all. Varaflow, I think all steps of Varaflow use um, very little memory. RAM
That's
memory
okay.
is not an issue. The thing is the number of threads, because you can scale it up. You can
Mhm.
ask for more threads, and then you run your analysis faster.
Okay. That's so cool. Okay, is another question here.
Mhm.
So, Bibiana said, very interesting talk. Thank you. There is an automated connected to Gizate to import and deposit sequences with ViraFlow.
So this is an interesting question. Thank you. In fact, Varaflow, the when you run Varaflow, you can generate a short summary. For example, that's the most required output, where you're going to have every sample with the lineage assigned, some statistics about the genome coverage, breadth, and depth, and so on. But Varaflow itself does not submit data to GISAID. Uh, what we do is that when we connect the viral flow output to the virus genome DB, which we are still developing, it's not openly available yet. Virus genome DB does that, because remember, to submit data to GISAID, we need to have all the other metadata associated, at least a minimum set of metadata. So that's a requirement for those sequences and information to be epidemiologically relevant. We need to have the data of sampling. We need to have uh, the a specific geographic location where it was sampled. So that's why we do it at the virus genome DB level. Virus genome DB does store this metadata, and then we import the data required for filtering and that to contain the statistics of the genome assembly, to filter out, and then virus genome DB filter out only the good genomes, the ones that are above the threshold, and then submit it to this.
Cool. Great. Thank you very much for the talks. And I guess we don't have any further questions for now, and we have to keep going to the short talk. So thank you very much for accepting. Thank you for showing this work. Still like amazing. Um, I had one more question that's not really related to epidemiology, but it's a curiosity for me because
Mhm.
I work with human data, and because we know that these viruses they can cause an effect in the human genome and sometimes changes in the
Mhm.
human genome. But you said that you don't track these things because of ethical reasons, and then you can't share the people's genomes.
Yeah.
But do you think it can be used, the sequencing data you have can be used in the future for looking at the effect that this, in the long term, this virus could have caused to the human genome?
So I think for the most of the data generated for genomic surveillance of those viral pathogens, nobody is able to extract meaningful genomic data. Meaningful, what I mean is that, well, let's say I want to evaluate or test the hypothesis if this virus infection really modulated or made any change in the host genome, because what we do, and that's something I forgot to add in the presentation, what we do most of the time, or for most of those sequence generators, is that we amplify the viral genome, right? So we amplify using primers that prime pairs that cover the whole genome, and then we're gonna end up with a huge amount of amplicons from the viral genome. But then we know that when we extract RNA or DNA, there is always some leftover of the host
Okay,
genetic material.
so it's like,
But this will be randomly selected, and I think it won't cover even, let's say, 10% of the human genome.
oh, okay,
Uh,
just a little. Okay.
yeah, I think for this strategy, that the most used strategy for genomic surveillance, it's not possible. But when somebody is using, for example, some metagenomic strategy where you deplete the host ribosomal RNA and sequence all the remaining, then you're gonna have a lot of genetic material from the host that is being sequenced together. But
Um,
for this, you're gonna need much more depth. We're gonna need
mm,
machines with much higher throughput.
mm,
So yeah, I think
yes.
evaluating those samples, the only way to evaluate from the host genome perspective is to reassess the sample and try to resequence.
Okay. Yes. Yeah, makes sense.
Mhm.
Okay. Thank you very much, and thank you for this wonderful talk again. We're gonna keep going to our symposium with the. We have three presentations, three short talk.
Mhm.
Um, yeah, that's it, people. We have already here with us the three people that are going to present, and thank you. Have a nice day. Bye. Thanks. Bye. We can move on with a short talk. So we have here Pedro. Can start with you. We have also Mariana because like we're doing in alphabetic order, but I think her microphone is not on and her camera. Okay, so we're gonna start with Pedro. Okay,
Okay.
yes. Mariana is trying to solve that. Um, okay, so Pedro, he has a bachelor in biomedical sciences at the Federal University of Triângulo Mineiro, and he's a master's student in bioinformatics at the Federal University of Minas Gerais, and he's gonna present his short talk called "Design of Multiple Epitope Vaccine Against Histoplasm Capsulatum Through Immunoinformatics Approaches." So, Pedro, when you ready, you can start.
Okay, thank you. Good afternoon. It's a great pleasure to be here. I'm Pedro Marques, master's student at the Federal University of Minas Gerais in the postgraduate program of bioinformatics. And today I will present my last research entitled Design of a Multi-Epitope Vaccine Against Histoplasma Capsulatum Through Immunoinformatics Approach. So first we need to talk about the Histoplasma capsulatum, the responsible for the histoplasmosis disease. The Histoplasma capsulatum is an intracellular fungus distributed worldwide, mainly in the Ohio and Mississippi River Valley, but also in the South America, South Africa, and Australia. So how can you contract the histoplasmas? When you explore one cave with beds or living near a chicken farm
Sorry,
or pigeons,
but can you put like full screen, please?
it's in full screen.
Oh, here is not appear full screen. And you can go down there in a button and like
Yeah, I clear it.
yeah.
Now
Oh,
it's
is
okay.
it? Okay, here's not appearing for screen. Okay, I think people can read. If they cannot, they can say something. But I don't know why it's not for screen. But okay.
Oh, I can try to share again my screen, maybe.
Okay, yes, maybe better.
Just 1 second.
Yes.
It's share full screen, and maybe
Okay.
now it's okay.
Let me see. Yes, now is it? Yeah. Okay, great. There's an
Sorry.
error from StreamYard, I guess. I'm sorry for interrupting. Can keep going.
Okay. So here I can try again the presentation. It's better.
Yeah, if you want to start again, you can start again. But now it's
Okay,
great. Everything's great.
thank you. So it's my last job, design of a multi-epitope vaccine against Histoplasma capsulatum through immunoinformatics approach. So first we need to talk about the Histoplasma capsulatum, the responsible for the histoplasmosis disease. So the Histoplasma capsulatum is an intracellular fungus distributed worldwide, mainly in the Ohio and Mississippi River Valley, but also in the South America, South Africa, and Australia. So how can you contract the histoplasmosis? When you explore a cave with bats or live near a chicken farm or pigeons, you can inhale the excrement, the feces of these animals, and the excrement, the feces of these animals have a lot of nitrogen, one element more important for the growing, for the metabolic system of the fungus. So you can inhale the filamentous form of the fungus in the air, and this leads to one respiratory infection that often leads to a systemic infection and the death in immunosuppressed individuals. So inside the host, the increasing the temperature turns the filamentous form of the pathogen into the yeast form. And the yeast form is the most pathogenic form of the fungus. Also, there are many other hosts in humans, such as dogs and horses. And in our objectives, first, we need to say there are no vaccines available and have a few treatments for systemic fungal infections, such as histoplasmosis. And the diagnosis, it's very difficult to do because the initial symptoms can be mistaken for tuberculosis or the flu. And nowadays, we have an increase in the number of cases of histoplasmosis promoted by the animal behavior change and the urbanization process, exposing the humans to the fungus. So our objective is to develop one multi-epitope vaccine based on 4 experimentally validated epitopes by Kishko and collaborators in 2021, and 4 vaccine targets for proteins identified by the reverse vaccinology by our group in this year. So the epitopes are the antigenic determinant of one protein, the minimal part of one protein that can induce, elicit the immune response. And we have 2 main kinds of epitopes. The epitopes that can bind to the MAC1 receptor and elicit the cytotoxic T lymphocyte, and the epitopes that can bind to the MAC2 receptor and elicit the helper T lymphocyte, the main responses against Histoplasma capsulatum. So in our methodology, we use the IEDB and the ABC PRED to predict the epitopes from the 4 vaccine targets for ATL and CTL epitopes and the B-cell epitopes for using the ABC PRED. We use the IC50 equal 50 and the percentile rank equal 1 to get the better epitopes to get the better immune response against the Histoplasma capsulatum. We use 2 different methodologies for the MEC1 receptor and 2 for the MEC2 receptor. We overlapped these 2 different methodologies to get the better epitopes. Then we overlapped these final epitopes with the B-cell epitopes from the ABC PRED. So we have in final the MEC1 and B-cell and the MEC2 and B-cell epitopes. We constructed the multi-epitope vaccine sequencing, and then we used the toxin PRED, AllerTop, Vaxigen, and ProTparam programs to predict the toxicity, allergenicity, antigenicity, and the physiochemical parameters of the vaccine. Then we use the collab fold to model the 3D structure of our protein, and we refine this model using the Galaxy Refine. Then we use the HDOC server to see the interactions between our vaccine and the TLR4 innate immune receptor. And we simulated the immune response in silico using the SimSim server and the interferon epitope. And finally, we use the SnapGene to in silico clone the protein into the plasmid of E. coli. Our first result is the epitope prediction filter and the overlapping. So in the first line, we have the number of epitopes from the 2 different methodologies of MEC1 and the 2 different methodologies of MAC2. We filtered this number of epitopes using the IC50 in the percentile rank, and then we overlapped the two different methodologies in one single epitopes, the final epitopes for MAC1 and MAC2. Then we overlapped again the final epitopes for MAC1 and the B cell, and the MAC2 and the B cell. So we have 23 epitopes for MAC1 receptor and the B cell, and 11 epitopes for MAC2 and B cell. Here are the sequence of our vaccine, and we have some amino acids linkers linking these epitopes. And we added the 4 epitopes from the experimentally validated work from Kishka and collaborators in 2021. And we use the HBHA protein from mycobacteria because it's an essential TLR4 activator that can induce the TH1 subpopulation pattern of helper T lymphocyte as an adjuvant. So we use the HBHA with the adjuvant of our vaccine. Here we have the structure of our protein from the collab fold, and then we refined this model using Galaxy Refine. So we can see by the Ramachandran plot that we have a lot of amino acids in favorable allo regions, comparing in relation to the collab fold structure. And our protein, our multi-epitope vaccine, is predicted to not be toxic or allergenic, and predicted to be stable physiochemically and antigenic by the Vaccine program. Here we have the interactions between the vaccine and the TLR4. So in red, we have the vaccine, and in blue, we have the innate immune receptor TLR4. And in the right, we can see that by the HDOC server, we have a lot of contacts between the two proteins in green and red. And we also check it, and we have four hydrogen bonds between these two proteins and one salt bridge. So the interaction is predicted to be stable and favorable. We also simulated the immune response using the multi-epitope and the adjuvant in 3 doses by the SimSim server. So our vaccine is predicted to generate a lot of antibodies such as IgM and IgG, generate a lot of B cells, and differentiate the helper T lymphocytes and the F and G words, and cytotoxic T lymphocytes. And also some cytokines such as interferon gamma. But the most important thing is that when we compare the multi-epitope sequence with adjuvant and the multi-epitope without adjuvant, we can see that the core of the immune response is based only in the multi-epitope sequence. So the adjuvant only promotes one better delivery of the vaccine, and in some aspects, such as plasmocytes and interferon gamma, the adjuvant contribute to the vaccine. Then we in silico cloned the protein into the plasmid of E. coli. In red, we can see our insert, our protein, and we cross-checked the immunity for dogs and horses. So our vaccine have 25 epitopes for humans that are also 25 epitopes for horses, and 10 epitopes for humans that are also 10 epitopes for dogs, for the MAC1 receptor. So in the near future, we can test our vaccine for humans, and then we can test too for the animals, for horses and dogs, to check the immunity for these animals against Histoplasma capsulatum. So the expected immune response for our vaccine, it's based on the 4 vaccine targets that we discovered in the reverse vaccinology in the last paper of our group in this year. And we predicted for these 4 vaccine targets, 23 epitopes for MAC1 receptor and 11 epitopes for MAC2 receptor. So 23 for the cytotoxic T lymphocyte and 11 epitopes for the helper T lymphocyte. We added 4 experimentally validated epitopes by Kishka and collaborators in 2021. And our vaccine is predicted to be safe, stable, non-toxic, or allergenic, but more tests in vitro and in vivo tests are necessary. So the expected immune response for our vaccine is based mainly in these 2 kinds of cells, the cytotoxic and the helper T lymphocyte. And we see that our vaccine can interact with the TLR4 innate immune receptor. So our vaccine have the potential to interact with this innate immune receptor, promote the secretion of a lot of cytokines associated with the TLR4, and then differentiate the MAC2, the helper T lymphocyte by the MAC2 epitopes into the TH1 subpopulation pattern of the helper T lymphocyte. And we checked in our vaccine that we have 13 epitopes that can promote the secretion of the interferon gamma cytokine. And the TH1 subpopulation pattern also can secrete the interferon gamma cytokine. And the interferon gamma cytokine is very important to recruit, promote the differentiation of cytotoxic T lymphocytes. And this cell, the cytotoxic, it's the most important cell because the cell can lead to death, the cells of our body infected by the Histoplasma capsulatum. So imagine if you have one infection in the lung, in the respiratory tract, with the Histoplasma capsulatum. The cytotoxic T lymphocyte is the main cell that can arrive in this in the lung, for example, and kill the cell infected by the Histoplasma capsulatum. So more tests are necessary in vitro and in vivo tests, but our results are very good, and we have one potential immune response associated with our vaccine. Here are my references, and I'm open to questions. Thank you all.
Thank you very much, Pedro, for your presentation. It was nice and good timing too. Everyone, any questions? Let me see if it the chat. Someone's questions in the YouTube. Okay. Yeah, there's no questions for now. Someone has one more question. One question at least. So I guess we can move on. And today there's not much people watching us. It is Saturday. They know it's going to be recorded. So
okay, thank you.
Yeah, thank you very much for your presentation. So we're going to go now to not be, we're already late. So we're going to go to the second short talk. I'm going to go to Mariana. Mariana, I think now is everything's okay. If you can allow, yes. Okay, we have a question for Pedro. Pedro is still here. Just Mariana, sorry, just go back here. Yes. So Pedro, there is one question for you. So what's the main problem or gap in knowledge gap that you have encountered in your work?
Yeah, it's a good question. When we work with fungus, we have some similarities with the eukaryotic cells, the human cells. So when we discover the 4 vaccine targets, we need to check if the epitopes have a similarity with epitopes from humans, proteins for humans. So we need to delete, to exclude many epitopes that have similarity with humans' proteins. Is the main difficult that we have in our work, but we pass off this problem very well. We don't have problems with this. Have a lot of methodologies available in the literature solving this problem in the last 3 years, 4 years.
Thank you very much. Yeah, that's the question. Bruno, also thank you for presentation. Now we can move on with Mariana. Thank you, Pedro.
Okay, thank you. Bye-bye.
Bye. Hi, Mariana. The microphone.
Hi, are you listening to me?
Yeah, you can share your presentation, or you want us to share? You send an email, right?
Yes, I did.
Okay, so we're gonna share here. Okay. Just a second. Here.
I can try here as well.
Yeah, just open. Okay, so now I have it.
Okay, I'll try here.
No, I have it already open. Let me just the slides. Mm-hmm. No. Mhm. Okay. Yes, you can start, and then we can.
Okay. Okay. So good afternoon, everybody. My name is Mariana, and I'm been doing my research called "Searching for Allosteric Mechanisms in Enzymes of Plasmodium falciparum and Plasmodium vivax through classical and quantum mechanical methods." This work has been done at UFRJ under supervision of Professor Pedro Pascucci. Next, please. So we start out talking about allostery, which is a phenomenon in which we can inhibit the correct activity of the catalytic site through the binding of a ligand or any molecule. So it can change the motions of the protein and then inhibit its process. This process has been studied by our group through different works, especially regarding falciparum 2 enzyme. Next, please. So, falcipains and vivapains enzymes are enzymes that are responsible for hemoglobin hydrolysis in the erythrocytic phase of the life cycle of both Plasmodium falciparum and Plasmodium vivax that cause malaria. They are papain-like cysteine proteases, which means in their catalytic site, the residues most important are cysteine, histidine, and asparagine. Vivapains and falcipain 3, they share 90% similarity. However, no 3D VP structures have been solved experimentally. Thus, we have to use in silico methods to model them and study their structure. In the image on the right, we can see the overlapping, the alignment of the structures of papain, which is the main enzyme of this family, falcipain 3, and catepsin K. Catepsin K is a human homologous of the falcipains and vivapains. Thus, we have to always keep in mind the importance of avoiding toxicity and trying to select better cavities for the inhibitors we are developing. Next, please. The main goal of this work is to explore allosteric mechanisms leading to the identification of cavities for inhibiting falcipains and vivapains through methods of classical and quantum mechanics. Next, please. Therefore, we start out with the structures deposited in the PDB repository of falcipain 3 that was used as the template for modules of 3BP for vivapain 2. The protein was protonated at pH 5.5. It was prepared in the CharmG server and running on the software NumD3, the simulations. The classical mechanics have been done in replicates of 2.4 microseconds and corporal temperature with 0.5 molar of concentration of Na, sodium chloride. We've done we've reached the obtainment of conformational clusters through Umber 22 software. Next, please. Hi. Oh, thank you. So through molecular dynamic simulations, we can obtain the data that's on the left side. We are observing on the top chart the RMCSD, which is value of quantitative value for the difference of a structure with a reference, which is the starting one. For each replicate, we can see a different profile. And on the bottom, on the chart on the bottom, we can see two main clusters, but three with a shoulder as well. On the center chart, we are observing this RMSD against RG, which is a data of radius of duration, which gives us the idea of the compactation of the compactation of the protein. The smaller the RG, the more compact the structure is. So we can observe more clusters. We've done also a quantitative evaluation to select the correct number of clusters. And we saw that the best values for Vivapain 2 were given for 3 clusters. This is important because we can obtain the most probable conformations in a dynamical system to analyze, for example, the opening of cavities that in a stable structure, in a fixed one, we cannot. So that was the idea. Next, please. As the methodology, so with those clusters in hands, we submitted the structures to the Cavity Plus server, identifying cavities on the protein surface. And then we checked the identity of these cavities to the human catepsins, applying a cutoff of 50%. Next, please. With that, we decided to explore the allostery with 2 main strategies. One is the mechanical allostery, in which the Cavity Plus server analyzes through normal mode analysis, comparing the correlations of these cavities to the catalytic sites, and it returns us an allosteric potential, the z-score of at least 0.5. The electronic allostery is a calculation of the electronic density using MOPAC's software. And later on, the primordial software was used to evaluate the reactivity of residues and their profile on identified cavities. We applied a 5 electron volt band from the HOMO and LUMO frontier orbitals. Next, please. As for the first strategy, the mechanical one, we obtained cavities with restricted identity with a z-score much higher than the minimum. For example, cavity A returns us 1.8 or 1.29. And the maximum identity to catepsin, human catepsin, was of 25.7%, which is a very interesting cavity to be explored. Next, please. Also, cavity B was identified, and with a much higher z-score, reaching 2.57, and a highest identity of 40%. Next, please. The quantum evaluation of the reactivity gives us the idea, returns the idea that the frontier band is located on the catalytic site, but it also is expanded to other cavities, and we can see the cavity C adjacent to the orthostatic site, in which we can access the catalytic cysteine and thus design a specific ligand that will be able to capture these electron and maybe avoid the catalytic capacity of the enzyme. Next, please. After that, we analyze the influence of the conformations on the electrostatic surface and the correlation of groups of residues, using the initial and the most representative structures from the molecular dynamic simulations. We use the PBS software and PDB2PQR to evaluate it at pH 5.5, and the Bio3D package in R to evaluate the networks of motion of those different conformations. Next, please. The electrostatic surface of EP clusters shows us that the different cavities, they have different profiles of electrostatic charges, in which the more red color returns us a more negative profile, and positive potential is given with blue. So this indicates that a ligand for each cavity has to have different physicochemical properties. Next, please. Regarding the networks of motion of the different conformations, we can observe that the groups of motion, they depend on the conformation of the enzyme, and the circles, they gives us the number of residues that are correlated and how they are correlated to other groups. And we can observe as well that residues from the allosteric sites, they are connected to the catalytic site through different routes. It's important because thinking about inhibiting them, they are not going to inhibit each other. Next, please. Concluding, we can furthermore, please. Okay. Regarding the conclusions and perspectives of this work, we were able to get representative structures from a dynamical environment and identify 3 different cavities with allosteric potential and with restricted identity to human homologous. One for mechanical allostery, the cavities A and B, and they are located in opposite sides of the protein with different electrostatic profile. The cavity C, obtained from the electronic allostery, presents reactivity residues that could be the targets for inhibiting the enzyme in cavity C through the location of these frontier molecular orbitals. As a perspective, this work is to do a virtual screening assay using the data bank Zinc 20. And for cavity C is to use a fragment-based drug design, aiming the inhibition, a more specific one that's capable to interact with the electron in cysteine. Next, please. Thank you very much.
Thank you, Mariana, for a great presentation. Your results very interesting. Anyone has questions can put in the chat, please. I'm gonna ask her. Okay, we have one question. So, Viviana, congrats for the presentation. They intend to publish the in-house Python pipeline or in a GitHub page.
Yes, we intend to publish it to make it available, but we are still optimizing it right now.
Okay, you're optimizing to publish. Okay, but you're gonna put in a GitHub probably.
Yes.
Okay. Another question here. So Elvira asks, the search for allosteric mechanisms can be very complex, especially in poorly studied organisms. What is the main gap in the methodology, and how do you think it can or will be resolved in the future?
I think it is to the experimental part of it, because we have to test it. We have to go to the lab, and test them. We are lucky to have collaborators that are soon will be able to test and to express the proteins and to do that um, prove
Look
this
for the direction, right?
capability, this relationship. Yes.
Okay. Yeah. Okay. Good. Thank you. And there's one more question here, so we can move on to the next talk. So, Andre, great talk. Are there specific directions you intend to pursue to experimentally test the interaction of the future candidate inhibitors? We already talked about it.
Yes, that's what we want to do. We want to go to experimental test because it's needed. You know that.
So, do you know what specifically you gonna do for the experiment?
So we usually is to try to incubate to try to test the kinetics of the enzyme. Usually that's the actually it's one assay that has been done by this group with phosphine 2, and we hope that they could do that with our group of proteins, because we are studying the phosphine 3 and vivapains 2, 3, and 4 as well. I was just presenting the results for vivapain 2, but we are exploring it.
Okay, thank you. Thank you very much. Okay, I guess that answered. I have further questions. Now we can move on to Wesley. Thank you, Mariana. So, Wesley, if you can share your screen, please. Hi, Wesley. Let me stop here the other presentation.
Okay, just a minute, guys.
I forgot to introduce Mariana a little bit, so let me just give a little short about her before I start. So Mariana, that present right now, she's a bachelor in biomedicine from the Federal University of Health Science in Porto Alegre, and a master's in biophysics at the Biophysics Federal University of Rio de Janeiro, and she's a doctoral student right now. So okay, now we can move on to Wesley. I'm gonna give him a short introduction too. So Wesley Juan, he is a bachelor in biology. He has a bachelor in biological sciences by the State University of Paraíba, and he's currently a master's student in bioinformatics at the University of Rio Grande do Norte. So his talk is I'm going to put here. So how to save a life. Mortality and lethality are associated with an antivenom access and temporal itineraries of bothrops and venom in patients. So when you're ready, you can start.
Okay, Raissa. Hi, everyone. Thank you for the nice introduction of myself and my work that is called How to Save a Life. Mortality and lethality are associated with antivenom access and the temporal itineraries of bothrops and venom patients. First of all, snakebite venom represents the biggest public health crisis that you have never heard of. And why this is true? This is true for some of reasons. Like every year, uh, almost 6 million of of people suffer with the accidents caused by these venomous animals, and half of this population comes to a hospital because they are in a venomous situation. And when the things get worse, at least 100,000 people comes to death. So, snake bite phenomenon is essentially a disease of the poor people because mostly of affected population live in some of the poorest areas of the world, like rural communities of sub-Saharan Africa, Asia, Oceania, and Latin America. They are basically smallholder farmers, their families, herdsmen, and displaced peoples. And because of that, these are the population particularly vulnerable to this disease. So, and what about the snake bite bird situation in Brazil? When we look at Brazil, we know that bottle snake bite venom is the main cause of snake venom, and annually, more than 20,000 estimates are reported. And despite free antivenom therapy is available nationwide for everybody, we have another issues like the temporal itineraries of these patients to come in a healthcare centers responsible for treat the envenoming patient. So in this work, we we be- uh, we looking for mortality and lethality caused by this group of snakes, and we observed how this could be correlated to itineraries temporal itineraries of these patients. So to do this, we look in Sinan database the information about snake bite venom data for bothrops species. So in this database, we look to number of cases per year and per region and state of the country, and we considered the years between 2112 and 2021. We also consider the ethnicity, the gender, and age range of the envenom patients. We look at also to the temporal itineraries of patients, and if the patients present local manifestations and complications and systemic manifestations and complications. Um, from this, we calculate uh, using these two equations, the mortality and the lethality, and the information about the healthcare centers for envenom treatment in Brazil was collected in the health ministry system, and our follow step was using the Vilipop code. That is a free code allocated in GitHub that take the address of these healthcare centers and transform in geocode information that was used in KGs to make um, our maps and to make the which can be visualize our data. So and our results was the in Brazil between the period of the study we observed the uh, 264 cases of venom caused by botrops was recorded by Sinan. And during this period, the largest number of cases was geographically concentrated in the northwest region. These for occurrence and incidence. In these regions, um, uh, I'm sorry, accumulated the most range of occurrence incidents for envenom caused by botrops. When you look at deaths, we we saw that 76 deaths was resulting from botrops species, and the situation was the same from death to occurrence incidents in this region. And in North and Northwest regions, concentrate more than half of the cases in which the patient's clinical conditions involve to death. So, when we cross these results with the antivenom therapy available in our country, we observe that in the north, in the northwest, a sufficient number of patients experience delays of up to 6 hours in receive appropriate medical attention following a snake bite caused by botryops species, and these contribute to a high mortality rates. If you look at this map, you can see that the pitch point of healthcare centers are more concentrated in the south region, in the southeast region, when we compare to North and Northeast and even Midwest. So this disproportional spatial distribution of healthcare centers responsible for the, the distribution of antivenom in Brazil uh, represent a big problem because in the regions that more need this antivenom, um, um, get less hospital with the therapy available. So we can say that in Brazil, the access to antivenom therapy could be made in few hours, but some regions could be in days. These are a result of a study, and we verified that in the northwest, there is a important part of this population in Venom that could take more than uh, 3 hours until 6 hours than people in South and in Southwest. For example, another student, this one of Cristino in collaborations, um, uh, I'm sorry. This study look at the some patients in Amazonia region could take until 96 hours to come in a healthcare centers, and they need most of the times to use different um, type of vehicles like motorcycle, airplane, ambulance, car, can, bus. So this is a big challenge to our public health in Brazil. And in conclusion, we we published this work recently. So there is a lot of information in this paper that can make us conclude that bottle snake bit venom pose a difficult public health challenge in Brazil. The most affected population are made for the poorest people that lives in remote areas where the access to education, healthcare information are scarce. And with this, we can advocate there is a urgent need for the decentralization of antivenom in Brazil, and we still believe the epidemiological surveillance of the situation of botulism venom in real time could help in the management and the control of the birth of the disease in our country, providing more dignity to thousands of Brazilians who live in the shadow of neglect. So this is my team where they helped me during the study in my undergraduate so thanks. I open for question, and these QR codes that appears in my presentation could be scanned, and you see more information.
Great presentation, Wesley. Thank you very much. If someone have questions, put in the chat, please. We can ask him. 5 minutes to answer questions. Okay. Um, any questions? Nothing so far. Okay. Yes. Okay. If I'm gonna just give a here little heads up for you. If someone asks why I'm talking, I can ask Wesley. So everyone, thank you very much so far. So right now we're gonna have a short break. Okay, there's a question here, so let me ask first, and then I'm gonna move on. So Wesley, do you suggest any approach to improve access to treatment in the North and Northeast regions where availability appears to be limited?
Yes, we believe that the centralization of the distribution of antivenom can help in the situation because if you can provide antivenoms where is most necessary, this could solve this problem because antivenom works and works well and are available for free in Brazil. What is incredible because most of country you have to pay for this.
Okay, thank you. There's one more question here. So it will be that unfortunately, due to large size, we have shipping problems. With bioinformatic or biotechnology, how do you think we can help? Is shipping of the antivenom?
Um,
Yeah.
um, how bioinfo or biotechnology can help it to
Help
solve
with
this
that.
problem?
Mhm.
I think the epidemiological surveys are important to monitor, and these actions like the decentralization could be uh, improve the distribution of the therapy for everyone, but um, we can work in tools with bioinfo or biotechnology that can contribute to the improve of the antivenom and to create more therapies because we only have antivenoms and serum, I'm sorry, to treat antivenom, to treat a venom for venomous animals. So bioinfo, biotech can help in provide more technology to treat a venoms.
Okay, thank you for this answer. Just another thank you. Amazing presentation. Thank you from Bruna. Thank you, Wesley. So, thank you, everyone. So now we're gonna have 10 minutes break, and then we come back at 4:50, and we have a little presentation about the RSG. Then we're gonna have the awards because our short talks were evaluated. And right after the presentation, we're gonna have award ceremony. And so we see you in 10 minutes, around 10 minutes, 12 minutes. Thank you very much so far. Be right back. Hi, everyone. We're back. A little late. Sorry about that. So, I didn't present myself in the beginning today. My name is Raíssa Feitosa. I am the actual president of the RSG Brazil. Now I'm going to give you a little presentation about the RSG Brazil, and after that, we're going to go to our award ceremony and our final collocations. So, let me just share with you. Okay, so what are the RSGs? Now, I guess the first important question you probably that are here and registered already know. Sorry about that. But there's some people that probably gonna watch this through YouTube and never heard about RSG. So RSG are a group of students that are came from the International Society of Computational Biology Student Council. They thought about creating groups around the world to help to share bioinformatics in different countries. So this is all the countries in the world that has RSGs active according to the Student Council website. You can see down here. So the RSG Brazil started actually in 2009, and then it ended for a brief period of time between 2011 and 2015. And then 2015, it opened again, and still working since 2015 until today. This is just the map of the world with the active RSGs. So right now, the current board is composed by these people. I'm the president, this vice president, Sofia, and the secretary, André, Thais, too, and the treasurer, Clara, and our faculty advisor, Professor Raquel Minardi. So this is just the distribution of members now around Brazil. We have around 20 members so far. So this presentation too is to talk about that we do calling for new members. I'm going to talk a little further, but now I'm going to talk about the active projects we have in RSG Brazil. So we have 6 active projects currently. The first one is Turning and Learning, in which we had a course 2 days ago. I guess some of you that is here at the Symposium participate on our course on molecular docking for leishmania, using leishmania as an example. That Vicente gave you the course. We also have a podcast in Portuguese language, and we have over 20 episodes. We have competition that probably some of you already know. That's the Brazilian Bioinformatics League. We have social media, and we share a lot of content on bioinformatics. We also have the Bioinfo outside of the bubble, Bioinfo fora da bolha. And we have the symposiums. This one you are now is the 7th symposium promoted by the RSG Brazil. So this is the active projects that compose RSG so in the moment. So the podcast, as I said, is available in any platform you can access. So all the platforms. So far is all in Portuguese, but we're planning to do some episodes in English too. So we have the competition, which happens during the X meeting, which is the biggest Brazilian Congress in Bioinformatics, promoted by the AB3C, the Brazilian Society of Computational Biology. So the last one happened this year in June. This is just some pictures from the 3rd edition that we had this year. In social media, you can find us in all the social medias. I guess you probably know too if you are here in YouTube. So you can find us on Twitter, on Instagram, Facebook, and we always share content about bioinformatics. The Bio Info Outside the Bubble, which is Bioinfo Fora da Bolha, is a project to try to reach a broader audience, more people outside the academic bubble. So we want to share bioinformatics with like our grandmas, grandpa, our family that never heard of it. And we want to talk in this project has the intention to talk in simple terms using an easy language accessible to everyone. And we also have the intention to go to schools in the future to share more bioinformatics and bring more people to bioinformatics. Training education, as I said, we had the course here. This is just a QR code for the course that already happened. We are continuing this week with course and symposium. And but if you, we have now opened a form in Google. So if you are available, access through this QR code to answer, to help us in producing new courses in the future. So we are planning to do some courses too next year. So if you have any suggestions, so please just fill this form and get in touch with us. We also have this form available in our linking tree that you can find on the Instagram page. This is the symposium, as I already said. So this is the past editions here. Down here, the past two were online, just like this one. But next year, we have the plan to do face-to-face during the X meeting. So stay tuned for our next symposium next year. We already also have a theme we're going to share with you as soon. So viewer, stay tuned. So this is just the last one in person that we had during the X meeting 2019. One is in bioinformatics and all the other editions. This is like the sixth symposium registration. So we have 500. This one was a little less people. Don't have the data here to show you, but we also have some publications so far. We have two that came from the LBB, which is the competition and the league that we have, the project of the competition. So we have these two publications. And now talk a little bit about how do we select members. So if you are interested in being a member of the RSG Brazil, if you are studying bioinformatics, study bioinformatics for a long time already, but you want to share, you want to help to with the community, you want to help to the awareness on bioinformatics and help us with the work we do. So we're going to open a call-in. We had one this year in April. We're going to have another one soon this year still. So we plan to do in the first week of December. So again, stay tuned to our social media for this. But we look for people that have a compromise, responsibility, because it's a volunteer project and we need a very responsible people to make our work, to make it work. So we also value a lot of proactivity and initiative because our project needs leads, some people to lead. We also appreciate a lot of creativity because we also can create new projects. If you have a project in mind and want to get together with other bioinformatics students and put and out there, just come join us. And also we look for group work is extremely important. We have monthly meetings. We are always interacting with each other, so nobody do anything alone at RSG. Dedication and follow the rules that we're gonna share in the calling that we're gonna make further. So if anyone here have any question about the RSG, about joining it, so please you can put the questions in the chat now. I can answer any questions for you. And so that's pretty much it. You also can reach us through our email or social media. And this is the QR code for our website for who doesn't have it yet. But I believe most of you do. So yes, if anyone has questions, I'll be happy to answer. Okay. Okay. So if you have any further questions, you can also ask us in the social media in the email. So I guess for now nobody have any questions. So right now, I guess the most important moment of the whole symposium, we're gonna do our awards. Let me just check here. I already have our okay. We already have a first and second place, like because we have the first place and the honorable mention, as you probably saw in our website. Um, just a second, everyone. Gonna go now to. Okay. Okay, now no more suspense, no more thriller. We're gonna announce. So, our first place. We did have we had 5 evaluations each of our work. So, our first place goes to Raissa Medina Santos. So, Raissa is a bachelor at the University FUMEC, and she did a master in the Federal University of Minas Gerais. So she won the first place. So congratulations, Raissa. We're gonna get in touch with you to give you the award. It was a very good work. So now we have the honorable mention. So honorable mention. Just a second. Okay, honorable mention is Mariana. Mariana won the second place. Mariana Simões Ferreira. She's a bachelor in biomedicine, the Federal University of Health Sciences in Porto Alegre. She has a master in biophysics. So congratulations, Mariana. You got the second best talk. Okay, there's actually there is 2 people has the same grade. Okay, we have something here, people, for the second place. Let me make sure here, because it seems like you have like 2 the same score. Okay, well, yes, we have 2 people with the same score. So we're gonna, I guess the fair thing here is to divide the honorable mention. So we have also exactly the same score. Oh, okay. No, sorry for this, everyone. People had they did a criteria to see because we had the same grade. Sorry, I was presenting here at RSG and then didn't see what was happening in the background. But actually, Mariana, you have the second place, so you got the honorable mention. But because we have this similar grades, we give the third place to Pedro. Pedro got our third place. So congratulations to Pedro. And Pedro just presented today too. I guess you saw it. It's design of a multi-epitope vaccine against histoplasma capsulatum. So congratulations for all the winners. It was wonderful to have you here. I guess very important to share about neglect diseases. We don't see many bioinformatics work on this topic. It was not easy to find talks to this symposium, just to let you know, because there's not much people working with both at the same time. But I guess we found like some of the best people that work with this to talk with you here. So thank you very much for making this happen, for registering, and be aware for our next symposium next year. I think we see you in person. Our next symposium will take place during the X meeting. So the X meeting is already announced. I don't know if you know, but it's going to be in Salvador, Bahia. And we're going to be there together and we're going to share the theme soon with you. Again, thank you very much for making this possible, because it's thanks for the people that are interested in bioinformatics nowadays that we keep our work going. So thank you, everyone. Anyone has some further collocations, questions? So I have some comments here. Yeah, great. Thank you very much, everyone. Do you have any questions? So you have a couple of minutes. Congratulations for all the winners, and thank you for your presentations and short talk. Please don't forget to put to fill the form with your email that you participated. So we're gonna register the presence of you here. So don't forget it. We're gonna close the forms in 1 hour. So we have like, if you have a delay, a problem with the internet, so we can still put it there. Thank you. Bye-bye, everyone. See you next year, the X meeting, face-to-face finally. Bye-bye.